Hi guys, my name is Cara and I'm working with Make Sense to bring you this video. It's just going to be about how to tackle certain questions on a paper like this, and we're going to go through the answers. Today we're doing an AQA GCSE Biology paper and we're doing the foundation tier. So I hope you're sitting comfortably and having a nice day, and we're just going to go ahead and get started. So question 1, we've got a multiple choice question. And it says chromosomes are copied during the cell cycle. Where are the chromosomes found? This is a fairly nice question to begin with. You should remember the answer is the nucleus. Just going to go ahead and put an X in there. And I've drawn a little picture for you here, just showing the nucleus. And hopefully this will help you remember where the chromosomes are found. So here we have the chromosomes, and here is the nucleolus. This whole thing here is the nucleus. And the cytoplasm surrounds the nucleus, and the ribosomes and the vacuole are found also outside the nucleus in the cytoplasm. So let's go on to the next question. What is the name of a section of a chromosome that controls a characteristic? And the answer to this, hopefully you know this, it's a genes, or just a gene. And it's important that you remember each chromosome has a large number of genes, and genes carry the information that determines your traits, which is essentially what this question is asking. Now it's given us a figure 1, and it's important you always read everything it gives you. So it says figure 1 shows information about the cell cycle, which is probably going to be important in the questions to come. Question 3. Which stage of the cell cycle in figure 1 takes the most time? Now this is one of the easiest types of questions you can get. It requires no past knowledge. You just need to look at the picture carefully. So let's go back. It's asking us which one takes the most time. So here we've just been given a circle, and we can see that how big the chunk is will represent how much time that phase of the cell cycle takes. So we can see mitosis is quite small, cell growth is a bit bigger, and copying of chromosomes takes the longest time. It has the biggest chunk. So we can just go ahead and put an X here and move on to the next question. During mitosis, cells need extra energy. Which of the cell structures provides most of this energy? Now, this question, if the answer should be quite clear to you. It's the mitochondria. The mitochondria are the powerhouse of the cell. Always remember that mitochondria equals powerhouse of the cell. Here's a mitochondria if you don't remember what it is. Here's a picture I put in for you. So we can just go ahead and put an X in here. And we'll move on to the next question. It says, the cell cycle in figure 1 takes 2 hours in total. The cell growth stage takes 45 minutes. Calculate the time taken for mitosis. This question also requires no past knowledge. It's just a maths question. And we're just going to take our time. I know maths can be scary, but as long as you don't overcomplicate the question they're giving you, you should be completely fine. Okay, so we're just going to take our time. It says, the cell cycle takes 2 hours in total. The cell growth stage takes 45 minutes. And all it's asking us to do is calculate the time taken for mitosis. Okay, so we're just going to go back to the figure 1. Now, what did it tell us? It told us that the whole thing, all the way around, took 2 hours. 2 hours total. Okay, and it also told us that this stage right here, that stage right there, took 45 minutes. Okay? So now we're just going to look at figure 1 and just figure this out. Okay, so these little notches on the end here, 1, 2, 3, 4, 5, 6, 7, 8, 9, we have 10. Okay? So we can see that we have 10 notches across the whole circle. Okay, now should we see how many copying of chromosomes is? If we just start here, got 1, 2, that's 1, sorry, 2, 3, 4, and if we take this little bit over here and just add it on here, we've got 5. So essentially copying of chromosomes is taking half of the time, okay? So we know that the copying of chromosomes is going to be 1 hour. 1 hour we've got. Let's just write that in here, 1 hour. So now we know copying of chromosomes takes an hour, half of the time. Cell growth takes 45 minutes. And all we've got to do now is figure out how long mitosis takes. So 2 hours total, we're just going to minus an hour, give you an hour, and then we're just going to minus the 45 minutes of cell growth. We're left with 15 minutes. Fairly easy question. Might just take a bit of time, a bit of thinking about, but just don't overcomplicate it. And for this question, it is essential that you show you're working. Don't have to write loads. It can be very simple. Just do 120 minutes minus 60 minus 45 equals 15 minutes. And also essential that you write 15 down here. There you go. That will get you both the marks. That's 2 marks you've got just on this simple question. So satisfied with that, we'll just go on to the next question. And we've got another picture. It says it shows some cells in different stages of the cell cycle. There you go, we can see that, pointing out 5 different cells to us. The question is, which cell is not dividing by mitosis? A, B, C, or D? So I guess for this question we can just ignore E, don't need to worry about that. And we'll just start with A, I guess, and see what it's telling us. Right, so you can see the chromosome starting to get pulled towards opposite ends of the cell. You should know that this is a part of mitosis. You can see that quite clearly in the cell. So A, it's not A. On to B. B looks like the cell is starting to divide. They're not quite 2 separate cells yet, but it looks like it's starting to divide. You can see the chromosomes in 2 little clusters. And so this is a part of mitosis. It's not B. We'll go into D. You can see quite clearly in D the chromosomes lining up at the metaphase plate, which is a part of mitosis. So the answer is not D. So we're left with C. And if you didn't see it right at the start, just looking at this, you can see that C doesn't look like it's doing very much, just looks like your average cell. Since it says figure 2 shows some cells in different stages of the cell cycle, it's clear that C will be an interphase. It's just growing and increasing the number of subcellular structures. This is not in metaphase, but it does come right before metaphase. So our answer, C. Next question. Cell E, now we're looking at cell E. in figure 2 contains 8 chromosomes. So we just go back and look at cell E quickly. Once again, doesn't look like it's doing too much. There we go. It says cell E divides by mitosis. How many chromosomes will each new cell contain? Right, so it's important to remember in mitosis, the cell cycle doubles the amount of chromosomes before mitosis occurs and the cell splits. Okay, so let's think about this in order. So, cell E has 8 chromosomes. This will then double to 16 in interphase when all of the chromosomes are doubled, replicated. And then when the cell splits at the end of mitosis to form 2 new cells, that will once again leave each cell with 8 chromosomes. Mitosis, it will just leave them, after mitosis they'll just be left with the exact same number of chromosomes. So our answer is 8. Next question. Why is mitosis important in living organisms? Tick one box. Right, so let's go through this. First it says to produce gametes. Now, gamete is formed in meiosis, and it's very important we carefully read the question. This is not about meiosis, it's about mitosis there. Okay, so this is not correct. To produce variation. In mitosis, the cell duplicates its chromosomes and it forms 2 identical daughter cells, so no variation occurs. The daughter cells are identical, so this is not correct. Next, to release energy. In fact, mitosis actually takes up energy. It needs energy to occur, so it is not this. And we're left with to repair tissues, which, if it didn't jump out at you straight away, we've just gone through this by process of elimination, and you'll come to the exact same answer. So there we go, to repair tissues. It's important to remember, if an answer jumps out at you straight away, you know that's the right answer, go ahead and put a cross in it, mark that question. If it doesn't jump out at you straight away, and you're not completely sure, I would recommend just doing the process of elimination. Even if you're left with 2, at least that gives you 2 answers that you think may be correct. That gives you more of a chance than just ticking any of the 4. Always do process of elimination. Go through which ones you know can definitely not be right. And it doesn't matter if you get it straight away, if it takes you a few minutes, you have to do this process. As long as you get the answer, you've done great. Okay? So we're going on to our next question. Right, we're going on to a different topic now, it looks like. And it says, plants are made up of cells, tissues, and organs. Draw one line from each level of organisation, to the correct plant part. Right. So, it says plants are made up of cells, tissues, and organs. Right, so we should know that cells make up tissues, and tissues make up organs. Okay? So, cells work together in a tissue, tissues work together in organs, and each organ has several functions. So, a leaf. Our first one. A leaf is an organ. Okay, so we're just going to go ahead and draw a line here. Make sure the line's very clear. There you go. A leaf has several functions. Next, root hair. A root hair actually is a root hair cell, and a cell is neither a tissue or an organ, so this won't be either of them. Spongy mesophyll, there we go, that's a tissue in the leaf. So our question is done already, but just to clear up the last 2, a vacuole is a part of a cell, not a tissue or an organ, and xylem cell gives you it right there, tells you it's a cell, so you don't even need to think about that one. Next question. Got another picture here, and it says it shows a plant cell drawn to scale. There we go. Where in a plant would the cell in figure 3 be found? Your options are epidermis, palisade, mesophyll, phloem or xylem. So shall we just go back and look at this once more? And you should be able to recognise from your textbooks or from your lessons, from any YouTube videos you've been watching, that this is a palisade mesophyll cell. Okay, you should be able to notice this just from this large vacuole sac in the middle. And surrounding this, you've got the cytoplasm, and in the cytoplasm you should see the nucleus. I'll just point to that for you. And you should see here the chloroplasts. Okay, so these should be a big giveaway. And here, again, this is called a sap vacuole, which should be a big giveaway. Okay? It's not the epidermis. Epidermis is actually the one with the stomata that opens and closes, and it's not phloem or xylem because they don't have that type of shape. So let's just put an X in here, and on to the next question. Right, calculate the length of the chloroplast labelled on figure 3. So for this question, you're going to need a ruler, and you're going to need just a pencil or a pen. This question will be a lot easier if you've got the paper in front of you, but I'll just talk you through how to do it when you have the paper in front of you, okay? So here we go. You'll just need a ruler, and you'll need to take this chloroplast length, here to here, and basically all you need to do is see how many of these lengths fit in here. So you can just, with your ruler, just see how many fit in there, okay? Or you can measure this with your millimeters, your centimeters, and then measure the chloroplast length and do it that way. But either way, all you need to figure out is how many chloroplast lengths fit in this longer length, the 50 micrometers length. Okay, and you should find, when you've got your ruler and your pencil, that the chloroplast length fits in it 8 times. So then all you need to do is let's go back to our working. All you need to do is 50 divided by 8, which will give you 6.25, okay? And then here, you just need to make sure again that you write 6.25. Very important in biology exams, you need to check the lengths the same. So here it's telling us to put the length in micrometers. We just need to double check that we've been working in micrometers all the time. And here we can see that we have. So that's all fine, don't need to do any conversions, that's perfectly fine. And on to our next question. It says, as a plant grows, new root hair cells are formed from unspecialised cells. How does an unspecialised cell become a new root hair cell? What we're talking about here is differentiation. So that will be the correct answer. So there we go. Differentiation is just the process of an unspecialised cell in the meristome region becoming a distinct cell type. So it's very clear that's what the question is asking you for here. If that wasn't clear to you, let's do the process of elimination. So we have metabolism. Metabolism is the chemical reactions that take place in cells to provide energy for vital processes. That is not what we're talking about here. Transpiration, you should know, is the evaporation of water at the surfaces of the spongy mesophyll cells and the leaves. And transport, I'm very sure everyone knows what that is all about, but in biology it's a molecular ion moving across the cell membrane or just moving in the bloodstream. So definitely not what we're talking about here. So that should leave you with differentiation. Next question. We're given another picture here, and we're just going to go ahead and quickly read what it's telling us. It says scientists can clone plants using tissue culture. Figure 4 shows the process of a tissue culture. So key words in this, maybe let's just underline them. Clone. We're cloning, we're using a tissue culture. And it's showing us, got a white flower, scalpel is removing some of it, some of the leaf. It's being put into growth medium, it's growing, and you end up with more white flowers. Fairly simple. Now on to the questions. Why might scientists want to clone plants? Let's start at the top. To create a new species of plant. Right. Well, we should know, if we go back to the question, we're talking about clones. Cloning is used to create an identical plant. An identical plant. There is no new species of plants created through this. So that's definitely incorrect. Second of all, to introduce variation into the plants. Once again, creating an identical plant, there will be no variation genetically. So that's not correct. To protect endangered plants from extinction. This sounds like a very reasonable answer, sounding like that is going to be the answer, but just double check or look at the last option it's giving us. To reduce disease resistance in plants. So we know this isn't correct because cloning actually increases disease resistance in plants, because you can obviously take a plant that you know and have seen is disease resistant to a specific disease, and you can clone it. So you're just left with a whole batch of disease resistant plants, resistance to this specific disease. So that's incorrect. So we go back to this one. Let me put this as our answer. Next question. What is the advantage of cloning plants using tissue culture? No special equipment is needed. Well, it's showing us that you need growth medium, you may need some other things. It's not showing you a lot, but it doesn't seem like that is going to be the answer. Growth medium can be seen as a special equipment. It's something you need. That may not be accessible to everyone. So moving on, plants can be produced quickly. That is seeming like it is going to be the answer, because as you should know, tissue cultures works fairly fast way of growing plants. Next, the flowers are all different colours. Well, the figure 4, in figure 4 that it's giving you, you can see it's showing you that you start with a white flower. It's showing you that you're just given more white flowers. There's no new colours introduced there. So a plain giveaway that this is not the correct option. And then the offspring are genetically different. They keep bringing this up, I guess, maybe trying to trick you. You just need to remember in the forefront of your brain, cloning just produces identical plants. There's no variation, they're not genetically different, no new species are made. They keep trying to bring this up, it's not correct. So we'll go back up here, plants are produced quickly, this is the correct answer. And moving on. The growth medium figure 4 helps plants to grow. Name one substance in the growth medium. This is just a question, fairly simple question, don't overcomplicate it. If you remember any substance that could be in the growth medium, you're probably correct. And we're just going to go through the answers. So one thing you can put is glucose or sugars. Second thing you can put is amino acids or protein. Third thing is hormones. The markers will allow named hormones like auxin. Fourth thing is ions or minerals, and you're allowed to put magnesium or nitrate, if that's something you remember. Number 5, you can put vitamins, and they'll allow named vitamins like vitamin B. The last thing you can put is water. And that is just an example showing you that water there. If you don't remember a specific substance, just put anything that you think could be a correct answer. Water helps plants grow. Even if you haven't been told that's in growth medium specifically, like hormones, like auxin and like ions, which I'm guessing you will have been told in class are in growth medium. When I was taught, I wasn't taught that water is in growth medium, but it is correct. So even if you're sitting there, your brain is stuck, you don't remember anything, just put something more obvious, like water, if you don't have anything else to put, because it is correct and you will have got a mark. Next question. A question like this, you just need to have memorized here. It says it shows the human digestive system, and you just need to have memorized this from your textbooks or videos of what the answers are going to be. Here you have a stomach in A, B is the small intestine, and C is the liver. Hopefully you will have remembered those. Next question. These questions are always quite fun. You just get to pick from this list of words here which word you think is correct, and you complete the sentences. So whatever you do, make sure that the answer you put is an answer that's in the box. Okay? So let's start. Digestion is the process of breaking down large food molecules into smaller molecules that are blank. So let's just go through the list here. Molecules that are catalyzed, no. Molecules that are denatured, no. Molecules that are digest, obviously not. Energized, excreted, ingested, insoluble, soluble. There we go. Soluble is the correct answer there. Soluble. And this is obviously necessary so that the food molecules can be absorbed into the bloodstream. There you go. Enzymes help break down food because they blank chemical reactions. The correct word here is catalyze. And this just means to activate and or speed up the reaction. And this is by the lock and key method you will have learnt about. And the last one, if the temperature of an enzyme gets too high, the enzyme is denatured. Is the correct answer there. And this is when the shape of the active site changes, so the substrate will no longer fit into it. This also happens if the pH gets too high. Okay? Protease is an enzyme. Protease breaks down protein. What is the protein broken into? All this question is asking you is what is protein made of? Like, what makes up protein? And the answer is amino acids. Amino acids make up protein. That's all it's asking you. It kind of distracts you with the protease, distracts you with a few things it doesn't need to say, but all it's asking you is what protein is made up of. Next question. Why is protein needed by the body? And you can have two different answers here. Either of them will get you the mark. The first answer is for growth. Obviously, protein helps you grow, which is why it's an essential part of your diet. And the next answer you could put is for repair / replacement. You can choose either there, for repair or replacement of cells / tissues. Sorry there, guys. / organs. So if you put for repair of tissues, there's your mark. If you put for replacement of organs, there's your mark. Gives you a few options you can put there. Both of them are correct. Next, just down here, which organ in the human digestive system produces protease? And here protease comes up again. This is just something you need to have remembered. But if it helps you, protease is an enzyme that catalyzes the breakdown of proteins in food into amino acids. Okay? And where does food get broken down? In the stomach, which is your correct answer. If you just know what protease is, you can work off that to try and figure out the answer. If you straight away know where the protease is made, there's your answer as well. Whichever route you go down, as long as you get the answer, you're doing great. Okay, next is the dreaded longer questions. But although it looks a lot more daunting than these multiple choice questions, the marks are not that hard to get. You just need to read the question very carefully, and you need to make sure you answer every single point of the question in your answer. Okay? So the question says, describe how you would test a sample of food to show it contains protein. Okay? And you also need to give the reason for any safety precautions you would take. Okay? To get a level 2, so we just quickly talk about how the levels. You have level 0, which is where you get 0 marks, sorry. You have level 1 where you get 1 or 2 marks, and you have level 2, sorry. Really confusing you here. You have level 0, level 1, level 2. Level 2 is the highest level on a question like this. And to get a level 2, so 3 or 4 marks in this question, you need to have reference to where you're at. You need to have a positive result and a reason. And you need to write about why a safety precaution is required. Okay? So the question is, describe... Oh, we've already been through the question. Okay, so I'm just going to show you a sample answer for this, okay? Here's your sample answer. First, grind up the food, and then add biorate reagent to the food sample. The protein in the food sample will turn the solution from blue to purple or lilac. As a safety precaution, wear goggles to protect eyes, or clean up spills immediately, because biorate reagent is an irritant or corrosive. Okay, where you've got purple, you don't need to write both of these options. You just choose either of them. Either of them will get you a mark. Okay, this answer will have got you 4 marks. 4 marks for this question. And also a quick note: you will still get the mark if you write CuSO4 or NaOH instead of biorate reagent. Okay, so you will have still got a mark. So, finish that question, on to our next question. Complete the sentence. Obesity can be caused by a diet high in... This should be a fairly obvious answer since it's given you the options of fat, fibre, minerals and vitamins. The answer is in fact fat. Obviously fat is essential in your diet, but too much fat can cause obesity. Next question. Complete the sentence again. Choose an answer from the box again. Obesity is a risk factor for skin cancer, type 1 diabetes or type 2 diabetes. If you think back and you remember, the answer is type 2 diabetes. Okay, and if we just go through the options, it should be fairly clear that skin cancer has nothing to do with causing obese. Having skin cancer will not cause you to be obese. It should be quite clear that there's not much link between them two. Then you're just left with type 1 diabetes and type 2 diabetes. And if you remember, type 1 diabetes you can have from birth, and is when the pancreas produces no insulin. The cause of this is still not completely understood, but type 2 diabetes is when the body doesn't make enough insulin or doesn't make insulin that the body can use properly. And this type of diabetes, type 2, can be managed through diet and exercise. So if you remember that little snippet of information, you should be able to get the answer. And we've come to the end of that set of questions. I will be making a part 2 where we go through the rest of the paper. If you want, you can just take a little break, or you can go right ahead to the next set of questions, whichever you feel is best for you. I hope that's been helpful.